Yeah, let's start.
So, Manvita, you missed the last session. Last session was about phylogenetic tree construction and analyzing the evolutionary relationship of the data. So, did you go
Yes,
through the
ma'am.
presentation? Yeah. Did you
Yes,
go through the
ma'am. I
presentation
got the
once?
output also, ma'am. I just checked
Yes.
the construction of the phylogenetic tree and have gone
Hmm.
through the slides, ma'am.
Okay. So you can just run the commands and everything. And if you have any doubts, there's a doubt session coming up after next session, I guess. So you can clear your doubts that day. Whatever you didn't understand, I can just go through that once again. Okay.
Yes, ma'am.
Okay. So today's session is diversity. In this session, I will do that once we are done with the generation feature table, is my screen visible?
No, ma'am. No,
Is it
ma'am.
visible now?
No, ma'am.
Now also it's not visible.
No, ma'am.
Okay. Is it visible now?
Yes, ma'am. Yes, ma'am.
Okay. So, as I mentioned in the last sessions, so basically, once we are done with the construction of the feature table and getting the representative sequence, then we can move on to the next steps. So now, from the, using the representative sequences, we constructed the phylogenetic tree by doing multiple sequence alignment and then converting into a code by using different indices and then uploading it into a web server where a phylogenetic tree is generated. So basically, you can create a rooted and an unrooted tree where you can check that how much and what is an evolutionary relationship that is happening between those species. So that was one application that we can do after we get the metagenome data. Now second application is we'll be studying about diversity. So there are two diversities, alpha and beta diversity. So for this session we'll be just focusing on alpha and beta will be covered in the next session. So in alpha diversity, what we are checking. So basically, if we go by definition, then alpha diversity refers to the diversity within a single sample. So that means that in alpha diversity, we are basically trying to look for the diverse organisms in one particular sample. That is in a pond, in a river, or in a room. This is not like comparing the samples which is given by beta diversity. So when we see beta diversity, it is basically comparison between two different samples. This is like you can use those words like it is intra and that is inter. So alpha diversity basically checks for both the richness and the evenness in a particular sample. So what is richness that I am meaning here? The richness is basically the total number of unique species that are present or features, because from our nomenclature we are using features as one of the term that is coming from the data itself. So it basically checks how many number of species are unique. Say if there are 10 bacteria present, so out of those 10 bacteria, are they all similar? Are they all similar? Or they have different subspecies or subgenus? Like they can be 10 different bacteria as well. There can be 5 different bacteria, but they're present in more number. So total is coming up to 10. So this is what it checks. So richness is basically it is counting how many different species are there. It is still possible that if you have 100 species ka pond, in that there can be only 5 species that are unique, and those species ka population is more. So that is why the total species count is coming to 100. So that number of unique species will give you the richness of the pond. Next is evenness. Evenness where we will be considering the distribution of the species, that is how much of each species is present in a particular sample. So you can say that if there are total 100 species, 100 total species, in which there are only five unique species and all other are populations. So in that five also you can see that if one is present only twice, fourth is present like 20 times, and there is one species that is present 50 times. So you will say that the species that is present like 50 times is more because it has a more abundance in that particular sample. So that is what we are meaning here from richness and evenness. So as an example, if we take, so when we are comparing our gut microbiome diversity, because that is what our moving picture tutorial data set is, between a healthy individual and a patient individual. So here we will check how the richness and how the evenness are getting differed when we compare these samples. And we have also taken samples from different body parts. So we will be generating plots of both richness and evenness by using different indices, where you will see that how different is the microbial community for different body samples. So now next is, why is alpha diversity so important? So basically alpha diversity will reveal the richness and the evenness that is present in a sample. So like it will tell about the health of an ecosystem. Basically we have to put it into that terms. So if there is any one microbe that is too much, that means that is not a good thing, right? It is try to take the nutrients that other microbes require. So those other microbes that are not able to grow because of one particular microorganisms, maybe those are required more in that area. Say soil. In soil, for the soil to become fertile, you need a particular microorganism. But because of the other microorganism that is interfering with the growth of good microorganisms, then that may cause a problem because the soil won't be fertile, and then you will not be able to cultivate it properly. So these all issues can be detected when you determine the alpha diversity. So you can detect if there is any loss of diversity, and you can also check if is there any high diversity. So obviously, if a microorganism is present in an imbalanced manner, like it is present in a very high presence, that means yeah, that is abnormal, right? Because there might be some disease or there might be some overexpression of something. That is why that microorganism is present, and it will not be considered healthy. It will have a competition with other microorganisms as well, but eventually it will start to compete with itself because its own microorganisms are dominating that place. Second is diversity. It basically tells about the stable system. So if there are more number of microorganisms and they're all diverse, then these microorganisms cohabitate together and they provide like good conditions, like better environment conditions, so that for the other species that are present in that particular area, they also can get the benefits of those microorganisms. So that and all is important. It is not always important that the microorganisms even that microorganisms content should be high. It is important that the environment should be diverse, and based on the diversity, the microorganisms number should also be normal. It should not be in an abnormal range. So you can use all this in microbiome studies, where you can do an environmental sampling and everything. Then you can do it in clinical research. This is not just based on a water or a soil sample, but you can do this in a microorganism research in a healthy individual and a diseased individual. You can take pancreas, you can check for the microorganisms present in both a healthy and a diseased liver. You will be able to find that there are certain microorganisms that are harmful and pathogenic and will be the ones who will be dominating in the diseased person. And you can check the diversity as well. So this is the main purpose behind us doing the alpha diversity analysis. So now there are key matrices that we basically know for alpha diversity, that is Shannon index, phylogenetic diversity. Then we have observed features and evenness. But when we run the command that we're using for KIME2, we'll be majorly focusing on two, that is Shannon diversity index and faith phylogenetic diversity index, faith PD. So when we move to Shannon index, so what is this Shannon index accounting for? So basically, when we run the command, so in the sample, Shannon matrix is basically a quantitative metric that is trying to account for both abundance and evenness of the species. I hope it is clear what abundance and evenness basically is meaning here.
Yes, ma'am.
Yes, ma'am.
Okay. So If in a sample the abundance is also more and the evenness is also more, that means that states that there are good microbial communities, and that to diverse microbial communities which are spread evenly. That means it's a good system. So that is the total measure thing that Shannon index measures, and based on that it gives the values. If the values are higher, that means the community is more diverse. If the value was lower, that means the community is a little less diverse. Okay, say sample A has many species evenly distributed. Sample B has few dominant species. So it will give a low Shannon for the few dominant species because the system is not diverse, first of all, and the system is not even because there are few species and that too are being dominant and the other species are not present only. So that will not be a favorable situation where a high value will be given. A high value will be given to basically that system that has evenness and that has richness. So we need so this is how Shannon diversity index will calculate and give the matrix. Second is Faith's phylogenetic diversity. From the name itself, we can understand that somewhere in this evolution will be included. So when we are measuring it with this particular index, so what happens is the evolutionary relationships are also considered. So when we calculate the Faith's diversity, I taught this in the phylogenetic tree also. So it will use the evolutionary relationships concept. It does not only depend on the number of species and its evenness, but it will also depend on how different these species are from each other. And how it will do is it will be calculating the sum of the branch lines that are connecting to them. That will be based on the phylogenetic tree analysis that we did for those species. So that is how Faith's phylogenetic diversity will be involved in calculating the values which can be interpreted further for the results. So if we get a higher PD value, that means it is a more evolutionary diverse branches. That is, the microorganisms that are present, they are diverse, they are evenly distributed, and then the microorganisms that are present, they are more evolutionary diverse. That means they are not very closely related, rather they are different microorganisms that are present in a particular sample. And if we say that the PD value is lower, that means they had a shorter branch length. That means there are sort of a similar species together, that is, they are closely related species. So one is like in a room, it is me, my friends, my college friends, my school friends, all of that in one room. And in another room, I have me, my sister, my immediate cousins. So you can understand, right, in which one will have a higher PD and which one will have a lower PD. So this higher and lower is basically depending upon the phylogenetic relationship here. So more the diverse, it will be a good score, and lower, less diverse, it will be considered as a less score. So now we move on to the commands. So when we move on to the first step, so basically first step is the core metrics analysis. So in core metrics analysis, the inputs that we are giving is C. First input is we are giving is for phylogenetic rooted tree dot QCA. Why are we giving this tree? Because we want to include that phylogenetic faith index so that we can generate the evenness and diversification with the context to phylogenetic analysis. Now, second input here we are giving is that is table.qza. You remember this table.qza we got from the feature table analysis. The rep6.qza we use in the phylogenetic tree to get a rooted tree. But now the table that had all the feature counts we'll be using here so that we can analyze the alpha and beta diversity. So next is sampling depth. So based on the last in the phylogenetic tree analysis or before that as well, we saw that sampling depth is one of a thing where we saw that how much sequencing, like for one particular feature, how many reads we are getting. So based on that, that is considered as depth. So here we are taking the sampling depth as 1103. So that means that the species or the features that have sampling depth of more than 1103 will be retained. But the ones that have the sample depth of 1103 and less, that will be removed from the analysis. So here we are putting a stringent filter because we only want ones those features that have a strong read support. That is why we are putting a sampling depth filter. Next is we are using the metadata file so that the biological relevance is also drawn. That is for our sample reads, those feature IDs should not be shown as feature IDs. Those should be shown as the name. That is from where we are getting those samples. And next is we are getting an output directory. That is a folder will be created that will be called as core metrics results. And in this folder, there will be multiple files that will be generated. It will have the alpha diversity files, beta diversity files, everything it will be there. So for our analysis here, that is for alpha diversity only, we'll do this. So what it does is, what this core metrics does. First it does is it rarefies the data. That is when you are trying to generate these diversity files. So the first thing is you are redefining and verifying the data. So in this, what happens is that when you have the data, all the data will not be normalized and not be equal. That is one of the main concept I told, right? That if one sampling depth is 1,000, another sampling depth is 5,000, those data are not comparable, because obviously the system will show that the one which is a 5,000 sampling depth is a more prominent one. But that will be like a false analysis that we are doing, because the other one that is lower, even though that is important to us, will be removed automatically because it has a lower sampling depth. So we need to normalize the data. That is, we need to bring all to the same level so that we can move forward. Next is we are computing the Shannon and the Faith index. So we will get certain files in this .qza files. We will be implying Shannon index and Faith index to those, and then we'll be generating a visualization files. And those are the result files. Now we'll move into more detail. So here the rarefaction that is happening is this. I mentioned, right? We are sampling depth we are putting. So this is our normalizing criteria. So the concept is, before comparing any sample, we should ensure that the samples are not samples are comparable. If the samples are not comparable, then it becomes a little difficult to analyze all of it. So here the problem, the statement that I've given here is, it's different different samples have different sequencing depth. The same thing. The sample A is supported by 50,000 reads. Sample B is supported by 10,000 reads. Direct comparison can never happen because it will always give a biased result. So this, we don't require this. We need to normalize this factor, and then we need to perform the further analysis. So what is the solution is, like the solution that we do here is rarefaction. Now subsamples all the sample to the same depth. That is, bring all the samples to 10,000 reads depth. And if they are not coming under that, then it will automatically remove the sample. That is what why we are setting a sampling depth here. It is going to remove the samples that have a lower sampling depth, and it will only retain those. And then it will become a little compare the results. The data will become comparable, and then we can directly work on it with the further analysis. That is, it will generate a core metrics result where the files for alpha and beta diversity will be there, and it will use the metadata also to put the biological relevance, and then we can further move on to the result, because now our data is also normalized, our metadata is also feeded along with that, and everything is in the same picture. And after that is, you're going to calculate the diversity matrices. So this is the core purpose of this step. So here we are trying to use, so I told you, right, when we run this command, it's not just alpha diversity that is getting not only alpha diversity matrix is getting created, but here both alpha and beta, everything is getting created, and later we have to sort them out and take only those files for which we need to find the alpha significance and the beta significance. So here everything will get generated, both alpha and beta diversity. For alpha diversity, it is using the indices Shannon and Faith PD. Shannon again for richness and evenness, and Faith PD for evolutionary. But for beta diversity, the samples and the indices that they're using is unweighted unifrac, weighted unifrac, and break versus. So unweighted unifrac and weighted unifrac I already explained properly in the last session where we studied about the phylogenetic tree. But I will also take it in the next session, but I won't go in much more detail. It will be much more detail. It will be like a brief thing. And break vertex will understand. So this is, we'll set apart right now beta diversity, and we'll go understand alpha diversity. So based on the evenness, richness, and the evolutionary relationship, the files, the significance file will be generated, and then we will get the output files. So when we did the core metric phylogenetic analysis, that is the first command. So in that, we are getting these files. These are the files that are important to us for alpha diversity. We are getting at least 10 files in that. But I sorted out the 4 files that is necessary for us for alpha diversity analysis. You can see Shannon, vector, QZF, faith, PD, observed feature, evenness. These are the all 4 indices that basically is there that is required in alpha diversity. Now we will convert these Shannon and Faith PD.qzf files into .qzv files where we can understand and analyze this data in a more visualization format. So now these files are used basically for the visualization part and the significance testing part. So first significance testing is after this step, after the 4 metrics analysis, we move on to the next step, that is the statistical testing. So one statistical testing will happen with the faith PD command, and another will happen with the evenness command. So after doing this statistical testing, what will happen is that you will generate box plots and all, and then it will also give you the p value as well. So it is going to compare the samples and the samples present in the same sample as well. So in the same group, it is going to compare that in an intra manner, and it will check for the significance level. So we'll be stating null hypothesis, where the hypothesis will be that all the null hypothesis will state that all the groups have same diversity, and then we'll put an alternative hypothesis that is at least one group is significantly different, and we are going to set these parametric tests. So here we are using for this particular setup, we are using a Kruskal-Wallis significance test. It's non-parametric test. It is basically used to compare more than 2 groups. So here it is basically checking for the median values between the groups, that is in the one sample itself, and that is applied here. And output values that they're giving, that is its stats and p-value significance. So basically we know that if p-value is less, that is the probability value. If it is less than 0.05, that means the difference is significant, and we can say that it's an alternative hypothesis accepting here, because the groups are significantly different. But if the p-value is greater than 0.05, that means there is not much significance, and the groups are generally similar. They are not very distantly related. Basically, they are like closely related. So now I'll put the commands for these. This was like for the theory part of the session. So now the first command that we have to run is this 4 metrics analysis. So as you can see here, it is alpha and beta diversity analysis combined. So we'll be just understanding alpha diversity part today. So I told you, right, after running, first you guys run this command. So I'm pasting this command in the chat box. So open your directory and run this particular command. Both Manvita and Chaitra, run this command and let me know if there are any errors or something, and run it simultaneously so that I can move on with the explanation as well. Okay?
Yes, ma'am. Yes, ma'am.
Do you need me to wait for a minute so that you guys can copy paste and run?
Yes, ma'am.
Okay.
Ma'am, does the command take a lot of time?
Yeah, it will take a little time because it has to run and it has to create so many files. All these files
I got
needs to be
targeted,
generated.
ma'am.
You got the output?
Yes, ma'am.
So basically, the output should be when you do list files, Can you share your screen once? Chaitra, is the command done running for you?
Yes, ma'am. Now it's done. I got.
Okay. Any one of you just share your screen once? Yeah. See, it told that all these files are generated. Now you do list files. So you can see, right, a core metrics result directory is created, right? Now please go into the directory.
Sorry, ma'am, I couldn't hear you.
Can you please go into the directory that is core metrics.results?
Yes, ma'am.
List. See, you can see we got so many files here. So these are all the files that are at the output of when we do the core metrics analysis. Here, the outputs that we are getting, they are all normalized and they are over a threshold. And now, see, you can see here we got the Bray Curtis distance matrix, Bray Curtis Emperor PCOA. All of this is beta diversity. And then when we see this evenness vector faith PD and all, this is alpha diversity. Jacquard index and unweighted, weighted, all of this is beta diversity. This will cover in the next session. So majorly the thing that is important here is evenness vector and faith PD vector. So these are the files that got generated for our alpha diversity. So these files will be converting into the visualization formats and will be significantly comparing the samples because of using these indices that we can get, and we can check that in our samples that we have taken, how many samples or how many microorganisms are significantly related and how many are not. Okay. So after
Yes,
this
ma'am.
command is done, now you move back to your original directory. No, just do cd space dot dot space cd space dot dot. Yeah. So you came back to the original directory. And I'm sending two more commands. Now paste those two commands and run it. Now in this, I have inputted the alpha diversity core metrics file. That is this one. I'm doing it for both the faith PD and for the Shannon index. So here the input for one is the faith PD file and one is Shannon index. And then the metadata file is obviously there so that we understand the biological relevance as well. That is, we shouldn't get the feature number, right? That big ID we shouldn't get. We should get the biological name for whatever sample we are doing. And then the visualization thing. So visualization things will also get stored in a folder that is core metrics results. And in that, inside that, the files will be generated as .qzv files. You will get generated, you two files will get generated. One will be faith PD group significance.qzv and one will be evenness group significance.qzv. So after this command is run. So see, you can see that one command has been run and it has already been saved in the uh, core metrics result folder as a faith PD group significance.qzv. And when the other is also run, it will also save the evenness one. So here we analyzed and we understood both of it. So in what these commands are doing. So these commands are basically the statistical part. That is, we are statistical testing here. That is, if our samples are significant or our samples aren't. So can you list your files now? See, you again go back to the core metrics result folder. See, before these files weren't there. Sorry, do list ls. Yeah. See, before there was no QZV file, right? But now you can see two QZV files, evenness group
Yes, ma'am.
significance and faith PD group significance. So now these are the two files that got generated. Don't look at the other files, just ignore that. That is for the beta diversity analysis. So for this, the .qzb file that got generated, these two files you can upload there on the web server and you can visualize. So now I will show you the files, how these files basically look. So when you open the evenness group significance.qzb file, so it basically looks like this. I'll just zoom it in a little. So you can see the samples, right? So there are gut samples, then there's left palm sample, right palm sample, tongue sample. So these are the samples that we took, and here it has plotted against the evenness. So you can see these box plots. So basically these are telling the outliers. This is telling the median part, the central line. So by looking at this particular thing, you can understand that the maximum evenness is for left palm sample, right? It is the highest. And it is well distributed as well. That is, it is a big box. So you can consider this as a good diverse sample that is present, followed by the other samples, that is right palm, and then tongue, and then gut. Because it is not that the evenness is not up to the mark where it should happen. So this is how basically you understand that which samples are more diversified inside a particular sample, and which one is basically telling that, okay, so which one is more diversified, which one is more even, and all of that. So when you come to the statistical analysis test, I told you the overall p-value for all the groups it is showing as 0.009. So this should be less than 0.05, and yes, it is. So we can say that overall, if we see that there are certain significant differences, all these microorganisms that are present in different regions, they might not be of the same origin. There is slight differences that can be seen. Now, after this overall thing, they have compared it pairwise. So when they compared the gut with left palm, right palm, and tongue, with all of it. So here you can see the p-value that is coming is when the gut is compared to left palm, it is coming 0.01. So 0.01 is less than 0.05. So gut and left palm show significant differences. That means the microorganisms that are present in those two particular places show significant differences and are basically diverse. Then if you see gut with right palm, it is again 0.006, that is still less than 0.05. So these samples are also significantly different. But when you compare the other samples, see gut with tongue, here the p-value is 0.2. It is way higher than 0.05. That means the gut and tongue somewhat had microorganisms that are somewhat similar, and they're closely related. And I think it does make sense, right? Because tongue and gut, they are related. Like they're present in the gastrointestinal tract, right? And obviously there will be certain similarities. Similarly, when they compared everything otherwise, all the results came very near to the like this one is nearby. So like, but still it is significantly not uh, good because it is less. Uh, and similarly, these are the way higher than 0.05. So you can just say that, okay, these two don't even considered as significantly different. They are mostly closely related species that are present. And this is like neck to neck that is present. So this is the alpha diversity that we got for evenness. So basically you got to know that the samples are evenly distributed or not. And for that evenly distribution, you are getting these statistical values. So I already explained the Kruskal-Wallis test that we are using here. It's a non-parametric test, and we are basically putting the null hypothesis and the alternate hypothesis and then calculating the p-value. And then we will be understanding that, okay, is it significant or not? The second is faith PD. Now here you can see along with the evenness and richness, it is also going to include the evolutionary relationship. Are they closely related or are they not closely related? Here you can see that this one has the maximum like coverage, like maximum PD shown, that is here, but this one has a higher median value. So they are at a bottleneck that, okay, both left and right palm. Even though right palm is a little more diverse, but this is majorly about the phylogenetic relationship. These two are a little lower because they don't have that close differential distantly related relations. So, and when we come to the parametric test again, here also you can see the p-value is way less. That means these species are not very closely related overall. They are farther related. So when you come and when you see here again, so when you see gut with left palm, you saw that the evenness is also a statistically valid. Here you can see the p-value is again less. That means the gut and the left palm have microbes that are not related to each other. They are different. They have differences. They again right palm you see 0.03. It should be less than 0.05, but 0.03 is way too near. That is like a bottleneck situation happening here. For tongue, you can see 0.2. That means gut and tongue have microbes that have similar evenness, they have similar distribution, and then they're similar. They're closely related species. They're not much different. And similarly, if you see left palm with tongue again, obviously it will give a very high difference because those microorganisms are going to be different because their functions will be different, their habitating environment will be different. So that is getting validated here. You understand the point. So if gut and tongue is coming different, then there's a problem in our data analysis because it shouldn't happen that way, because we know as a fact that gut and tongue will have something in common. And similarly, when we do right palm and left palm, there should be not much significance because obviously the microorganisms relationships, their evenness distribution, their evolutionary relationship should be similar because they are like same, you're taking from the same sample. It's just one is a left palm and one is a right. So here you can see a vast differences between when you compare gut with palm, sorry, gut with palm, gut and tongue with palm. So, but it shouldn't be like palm with palm and tongue with gut. That shouldn't show much significance, and that is what it is getting corrected here as well. And Q value is nothing, just an adjusted P value. That is, instead of this, you put more stringent criteria to get these sort of P values. That's the only. So this is how the alpha diversity analysis looks, and this is what it tells. So is it clear?
Yes, ma'am.
Yes, ma'am.
Okay. So alpha diversity wasn't that big of a topic. It was short topic only. So that is why I wanted like it could have been covered faster. So it basically tells Shannon index. It basically does the faith index where we are comparing evenness, richness, and evolutionary relationship when we combine both of them. And this is how the results come. And then to just statistically validate them because we are dealing with numbers here. And then if the statistical validation is along with our understanding, then it is good. Here the results are good because this is a tutorial thing, so obviously the result should have come appropriate. So you can see that tongue is a little less significant when it comes to gut, but it is more significantly different when it comes to left palm or the right palm. Do you understand that?
Yes, ma'am.
That is it for today's session. Alpha diversity was not a very big topic to cover, so if you guys have any doubt, you can just ask me. Any doubts?
No, ma'am.
No, ma'am.
Okay. So, and I just needed a short update on your log files. Are you guys making your log files?
Yes, ma'am. We are doing.
Yes, ma'am.
Okay, and good, good. I think that's it for today's session. If you guys don't have any doubt, then maybe we can end the session.
Yes, ma'am.
Okay, ma'am.
Bhavani, are you there?
Ma'am, the Friday class will be about beta diversity right now.
Yeah, it will be about beta diversity. I don't think I'll be able to complete the entire thing in 1 hour. So maybe it will be a little longer session. So you guys have to be prepared for that. Okay.
Okay, ma'am. On Monday, we will have the documenting session, right?
Yes. Come again,
On
Mandita. I
Monday, we
didn't.
are going to have the documenting session right now.
Yeah, yeah. And if you guys have the thing is that I was planning to teach how to construct an engine, a UPGMA tree that day. So basically, that is like on paper. So basically, for you to understand how basically routing and unrouting works. So like if you guys don't have so many doubts, then maybe I can take that session. But if you guys have like a lot of doubts, you let me know beforehand. Otherwise, the session will go a little longer. It won't be for a 45-minute session. It might go for like 1.5 hours or something. If I bring up the topic of UPGMA and NJ, you just guys talk among yourselves and let me know.
Yeah, sure, ma'am.